Welcome everybody to the second of two sessions on our invasive plant species track. I'm Doug Johnson with the California Invasive Plant Council and my co-host.
It's Jutta Berger, also with the California Invasive Plant Council. Thank you for joining us, and we're happy to have here a suite of case studies that will highlight successes in stewardship and in restoration in invaded landscapes. Our first speaker is Emma Havstad. Emma Havstad is a senior restoration ecologist with River Partners. It's a nonprofit organization whose mission is to bring life back into rivers by creating wildlife habitat for the benefit of people and the environment. She has a bachelor's from Cornell University, a master's in conservation and restoration from UCI. Emma.
Thank you, Jutta, for that introduction, and thanks to everybody here for joining me with your Friday afternoon to talk about invasive and native plants. I'm excited that I get to share with you a project that River Partners has been planning for the past couple of years, and which will soon be moving into the implementation stage. Can you guys hear me okay? Yeah, great. As Jutta said, River Partners is a not-for-profit organization. We were founded in 1998 by two farmers in the Central Valley who saw an opportunity to address the crisis of habitat loss by applying large-scale agricultural techniques to habitat restoration and essentially farm riparian forest in the northern Central Valley. Since then, we've expanded into many watersheds across California, including most recently into the Alamo River in Imperial Valley. So first, let me orient you to the project geographically. The area that I'm focusing on is about 550 miles southeast of us here in San Jose. The Alamo River runs north from Mexico across the incredibly productive ag fields of Imperial Valley and drains into the Salton Sea. About 12 river miles south of the sea, the California Department of Fish and Wildlife owns 2,000 riverside acres that they manage for wildlife. So out of curiosity, can I see hands of anybody who's ever visited this parcel? Oh, we have a few. Great.
Not the parcel, not the parcel.
Okay. Okay, great. I'll take it. Great. Anybody else, if you happen to be in the area, check it out. It is actually one of the oldest properties, one of the earliest properties that CDFW acquired when they were starting the refuge system in 1931. Right. And it was at the time a very popular duck hunting spot. In the decades since then, unfortunately, the resources to manage and maintain the property have been somewhat lacking, and the current land managers now are facing a pretty extensive infestation of the most successful invasive tree in the Southwest, tamarisk, which you see in the picture on the left here. There's also been sort of an increase in use of the wildlife area for illegal trash dumping, so people taking out their old washing machines. I've seen piles of chemical bottles or just household trash and tires, and so it's unfortunate. But when we got with the land managers, our team came up with these 4 priority goals for the restoration of the site. The first is to create high-quality, self-sustaining habitat for both rare and common wildlife, particularly neotropical migratory birds and pollinators. Second is to address climate change by sequestering carbon, creating temperature refugia, and increasing connectivity. Goal number 3 is to create employment and recreational opportunities for the surrounding communities and really try to turn this back into the community asset that it used to be. And the final goal was to create a model project for broad regional replication. So hoping that what we learn from this first few phases at the wildlife area, it's actually called the Finney-Raymer unit. So if you hear me say that, that's what I'm referring to, the land that was outlined in the previous map. And there's potential for applying what we learn here to many riverside lands along the Alamo River as well as the New River, which runs in a similar course from Mexico up to the Salton Sea, but just to the west of the Alamo River. All right, so in our initial site assessment, we identified 2 distinct existing conditions that represent good opportunities for horticultural restoration. The top picture on this slide is showing the very dense and very dense infestations of tamarisk, which occupies about 600 acres right along the river corridor on the Finney Raymer unit. On the other hand, the site also has about 500 acres of riverine terraces that were cleared for agriculture a very long time ago and remain almost entirely unvegetated, which is what you see in the bottom picture there. And both of these conditions do also contain occasional native tree species. So the tree on the right of the top picture, I believe, is a black willow, one of 10 or 15 that are left on the site. And then the bottom picture on the horizon, you can see fairly robust mesquite. So that's a good early indication that soil and water conditions can support those species once they do get established. If we zoom in a little bit on this southernmost section of the wildlife area, this is a sequence of historic imagery showing the growth of tamarisk across this constructed wetland. It was actually planted with natives in the late 70s as waterfowl habitat and is now almost entirely tamarisk. Of course, we can't really discuss the dynamics of invasive or native vegetation in the system without looking at hydrology. The water of the Alamo River is almost entirely the Colorado River water, which is transported to the Imperial Valley via the All-American Canal, applied to the farm fields there, and then drains off those fields into the Alamo River. So the green lines on that map are showing the agricultural drains within the wildlife area, and there are many, many more of them across the reach of the river. These drains are, many of them, controlled by remotely operated gates, which open to let water through and prevent upstream flooding of the farm fields, which is just to describe to you how managed the water is here. So the hydrograph on the left is not really tied to rainfall or snow melt that you might see in a less regulated system. There's really not much variation across the year in the stream flow of the Alamo. That means that we don't really see much flood scour or sediment deposition on the floodplain, which would be required for our California native riparian trees like willows and cottonwoods to germinate, establish, and outcompete tamarisk potentially. So what that indicates to me is that we can't necessarily expect to just remove the tamarisk and see willows and cottonwoods spring back up. We're probably going to have to do some active planting to get them established there. While we were in this planning phase, we also had the opportunity to observe and document the fire dynamics of the invaded riparian corridor when about 7 acres of the project site burned in an accidental fire, which is actually quite common along both the Alamo and the New Rivers. So the tamarisk leaves and dead branches accumulate debris. There isn't flood flow that would remove that debris off the site. And so it creates conditions for intense and frequent fires. We thought we'd use this opportunity for a small field trial and see if we could get willow and cottonwood cuttings to establish in the newly burned areas without doing any watering or trying to ameliorate the salty soils in any way. So what we've seen is that, yes, a small proportion of the willow cuttings can tolerate this pretty harsh environment. However, the conditions are also very good for germination, new germination, and resprouts of the tamarisk. So in the September 2022 picture, you can see my one remaining willow is just about to be overtopped by tamarisk. So we learned two main things from this small field trial. First was that we are, not surprisingly, going to need to provide some herbivory protection, so we lost some cuttings to critters. And secondly, that we can anticipate needing to treat the tamarisk consistently and utilize all the tools in our toolbox, including the careful and judicious use of herbicides, because when there's this level of biomass regenerating from fire or mechanical control, we just don't expect those techniques alone to provide the outcomes that we're looking for. We've also been fortunate in the planning phase of this project to have a nearby model site that's a bit older that we can visit and learn from. About 60 miles northeast of the Finney-Raymer unit, we've got a 55-acre restoration site on the Cibola National Wildlife Refuge, which was completed in 2011. I turned off the water and stopped doing any maintenance in 2011. And it's been just a complete joy and treat to go back and see how the vegetation is doing. We got some grant funding from the Bureau of Reclamation to monitor wildlife response to that. That research is ongoing. We're about halfway through a year of monitoring that, and that's what that wildlife camera picture came from. That's a badger and 3 coyotes. That'll probably be in every presentation I give for the rest of my career. But yeah, really rewarding to see the really densely established mesquite forest here doing a great job. There's barely any tamarisk there. One of the speakers in this session yesterday called it passing the O test for obvious results. And I think this is my version of that. Feels pretty clear that the mesquite forest is doing what we wanted it to do. Although we love to get more data too, but to me it's passing the obvious test. And so this is kind of what we hope to recreate on the Alamo River at the Finney-Raymer unit. Soil conditions are fairly similar, and we're seeing indications that we could repeat this. Of course, when you're talking about tamarisk removal in arid lands, there's always a lot of interest in water use and potential water savings. So we asked our friends at the Irrigation Training and Research Center out of Cal Poly San Luis Obispo to quantify, using our older model project, their expectations of evapotranspiration at the older restoration site as compared to vegetation that was there previously and as compared to adjacent vegetation, mostly tamarisk. They use Landsat data to do this. Don't ask me the details of the metric model. I could look it up for you, but what you see in the results here, the blue bar is our restoration site. And so what we see by 2016, it looks like water use of the mesquite forest is sort of leveling off, and it looks like it's lower than what was there before we did the restoration. But we'd really rather get some newer imagery and analyze that before we draw any conclusions. We had a second site that we were trying to analyze as a comparison, but it also burned. And when vegetation burns, then the water use goes down dramatically at that site. And so it's not really able to draw good conclusions there. I think there's an interaction effect with fire frequency that I haven't fully developed. Last but not least, we did start collecting soil samples across the Finney-Raymer unit in order to refine our understanding of high salinity areas so that we can plan accordingly and pick appropriate plant palettes. We haven't collected sufficient within the tamarisk area to have any real relationship between infestation of tamarisk and soil salinity. But I suspect as we start implementation and moving into each field, we'll continue to collect samples and kind of refine that, and maybe we'll have more to share about that later. But the big take home here is, there are places at the Finney-Raymer unit with extremely high surface soil salinity, and we will need to adapt our plant palette accordingly or work on soil amendments to address that, which is something I'll talk briefly about in one of the next slides. But in general, the conceptual plan shown here, there's 2 sets. On the left is essentially the treatment type. So on those riverine terraces that are unvegetated, we're expecting we could start planting mainly mesquite forest pretty quickly. And in the other areas that are infested with dense tamarisk, we're planning to do 3 years of tamarisk removal and really retreat it pretty hard before we get into a planting phase, so that we have good control on that first. And then the map on the right shows our tentative plan for now of vegetation. As I said, I think we're planning to do this in phases, so we'll kind of learn as we go what does well and adapt that map. But that's our conceptual plan. And I am very happy to share that our phase 1 project has received grant funding, and so we'll get started soon. That will include these 3 fields in this map. Each phase is designed to include more acres of planting than salt cedar removal, so that we're not losing acres of habitat. And we're anticipating in this first phase to include field trials, including the soil amendments that I mentioned briefly. That include incorporating mycorrhizae into the planting plan, and some trials of gypsum. We're planning to collect high-quality data on costs of different treatment methods for tamarisk. We're hoping to update that metric model on evapotranspiration, and I think that there'll probably be opportunities for trialing different species directly seeding and measuring how much irrigation we feel each species needs. I'll probably think of more. Actually, breaking news is that phase 2 has received funding too. So we're gonna have a lot of work in Imperial soon. 1 minute, thank you, perfect. I'm gonna end by saying thank you to all of our amazing partners. There's really quite a few people and teams that contributed to this. The California Department of Fish and Wildlife deserves special thank you for having the vision of wanting to transform the Imperial Wildlife area, and for providing our funding so far. And with that, I will open it to questions if there's time. Go ahead.
Why you use gypsum as soil amendment? You have heavy clay inside.
There are some places of heavy clay, but it's mostly to try to reduce the salinity there.
Okay.
Where's the tamarisk beetle?
I don't know. Let's get it. It has it. It is there?
Yes, it's moved down past the wall all the way down to Yuma now. So
Okay.
it should be in the area.
All right. I'm ready for it.
The northern area.
Probably an obvious question, but do you anticipate needing volunteers for this?
I would love volunteers. I also expect that we'll be hiring. We have positions open. Check out our website, riverpartners.org. But yeah, I would love volunteers too. Yeah.
I lived there for 2 years, so I'm getting familiar with the area. So anybody working there should wear a face mask because it's some of the worst pollution in the air. I don't know if Alamo's is as bad as River, but it's the most disgustingly polluted river in the United States under black slime. And water rights can be hard, so I'm glad you're trying to do something there, but it's been hard hit. And people in the area tend to be really conservative, so sometimes didn't want to work with environmental people lost.
Yeah. Yeah. Go for it. Thank you. We're concerned about the soil salinity and tolerance for willows, but we are hoping that there will be areas, as we do, as I say, more detailed soil sampling where we identify willow. Yeah. Go for it. Feel free to cut me off if we're out of time.
That's the last question.
Okay, last question.
I was wondering what the survival rate for the cuttings was.
1 out of 100, but those they didn't get any water. We just put them out there to see what would happen. Yeah.
from Imperial Valley to the San Francisco Bay Area, we have Eric Wrubel, who is a biologist and vegetation ecologist for the National Park Service at the Golden Gate National Recreation Area, where he works on endangered species recovery and restoration. All right.
Thank you, thank you. I'm always amazed at the scale of work that River Partners does, so kudos to you on that. I'm going to be talking to you about another case study of a grassland restoration that we've been working on at Rancho Corral de Tierra in the Golden Gate National Recreation Area. So a little background. This restoration was initiated to protect Hickman's potentilla, Potentilla hickmanii. It's a beautiful small forb that's only known from 2 places in the world. The Monterey population is around less than 60 plants, and population in Montera, where Rancho is located, contains typically over 5,000 plants. It was discovered in 1995, and it really boosted the chances of this plant persisting and not going extinct. And it was one of the main drivers to the acquisition of this property as a national park. So that's a map of Rancho on the left side. And it's located in Montera, California, right near the coast, at the base of Montera Mountain. So there's a 2,000-foot gradient elevation from the sea to the top of the mountain within 2 miles. It's a biodiversity hotspot. So for our project, since the land is now protected for this population of Hickman's potentilla, we're working on other ways that we can help this plant out. And so we're looking at invasive species, which are one of the main threats to this population, specifically control of harding grass, Phalaris aquatica, Monterey pine, Jubata grass, and blue gum. We wanted to control these species that are encroaching on Hickman's potentilla populations, which you can see there in the yellow on the map on these spur ridges which come down from the mountain. The tan area is a large monoculture of harding grass, which is shown in the picture down there. It was planted as a pasture in the 20th century. So we wanted to control the harding grass mainly, and that's the portion of this project I'm going to talk about. Also, we'll mention that there's a serious conifer encroachment coming from Montera into grasslands there at Rancho. Another main goal for this project was to restore coastal prairie, which is an important resource for Golden Gate National Recreation Area. Coastal prairies are iconic of coastal California, biodiverse, and there's not very much coastal prairie left due to development and agriculture pressures. Also threatened by climate change and ORV use, and invasive species. A little more about coastal prairies. They're great carbon sinks. So that's a picture of a California oak grass with its roots extending 2 meters down. And the illustration shows that, by contrast, invasive annual grasses really have a lot less root mass, sequester a lot less carbon, support less productive ecosystems. So For the first phase of our project, which was spearheaded by Susie Bennett, who's here, and can correct me if I get anything wrong, we wanted to control the harding grass in the footprint that is shown here. We used herbicide to get rid of this grass using trailer booms and backpack sprayers on about 5 acres. We also did extensive collection of grass and forb seeds and began contracts to plant container plants. Also, then the entire area was mulched with rice straw grass to reduce resprouts of harding grass and control weeds and erosion. In 2017 and through 2018, we entered into a contract to amplify grass seeds at S&S Seeds Farm. The first harvest was collected in 2017 and direct seeded into strips of native grass using a couple different treatments, raking in, just broadcasting onto the ground, broadcasting on top of mulch. This was fairly successful for one of our grasses, Elamus glaucus, and then monitoring began. So the initial phase of monitoring used line intercept transects, 4 lines at 50 meters, randomly placed in the harding grass treatment area, and we recorded all plants we hit at every quarter meter. So results from this phase of the project. On the x-axis, you see the year and species of invasive grasses. On the y-axis is percent cover. What you see here is the green bars are harding grass cover, which went down significantly during the treatment period. But what you also see is on that yellow trend line is a dramatic rise in rat-tail fescue, Festuca bromoides, an annual European grass that's invasive in coastal grasslands. So we had excellent control of harding grass, but like a classic competitive release of rat-tail fescue. Native grasses also moderately increased during that time. Interestingly, California brome increased in cover by 14%, but that wasn't one of the grasses that was planted during that time. It was just, is that right? I think that's right. Then you also had an increase in Elamus glaucus. So I think California brome is a good grass to go with in the future here. And we didn't have much for purple needlegrass or California oakgrass. Then there's a little gap in management, and I took on this project in 2019. We received our second shipment of seed, so we now have hundreds of pounds of native grass seed, which is pretty cool. We did some tilling and planting. We also had a lot of annual grass to contend with. And you need good seed soil contact for plants to grow, as we all know. So we went to the herbicide again to control the annual grasses. We pioneered the use of the mantis tiller for making furrows in the grasslands. Seed drills are very popular and pretty effective at grassland restoration, but this site already had native plants planted in it, and it really needed something like less not as industrial of a scale as a seed drill. So this is what we found worked for digging furrows to plant grass seeds. This says we planted at 6 to 10 pounds per acre. That's wrong. It was more like 40 pounds an acre, so sorry about that. And then birds came, and they ate all the seeds out of the furrows. So then we tried to plant the seeds again, and then the birds came again. So then we bought some agricultural row covers and installed them, and that stopped the birds. We also found that supplemental watering was necessary because this was also a period of drought. So we really like our water tank. And we started trying a new monitoring method, a grid quadrat layout to improve detection of spatial patterns in the grassland. Because the location of different species is very patchy, and it's hard to get a grip on it on the ground. So we laid out this grid and went to all these spots and monitored every plant at every point in a 1 meter radius and estimated visual cover of those plants. And here's the results from the recent monitoring. Annual grasses or non-native species are the bars in yellow. This is the years 2020, 21, and 22. It appears that non-native cover is slowly going down, and native cover in green is slowly going up. I haven't put p-values on this, but I don't think it's statistically significant yet. There's a lot of variation. We still have a pretty big problem with rat-tailed fescue. So these are the dominant invasive grasses on the site, and you can see the yellow bars show rat-tailed fescue. It has gone down in the last 3 years, but not by much. And also you can see from this that that grass is our biggest problem on the site. The other invasive grasses are at low cover, including harding grass. Native grass cover overall is creeping up, but it's not super high. Doesn't get the O factor yet. But what's heartening is the frequency. So the cover is not high of native grasses in the plots, but we typically have 1 or 2 sprouts of native grasses, and these are our 4 native grasses that we're tracking: California oatgrass, Bromus carinatus, Stipropulchra, Elamus glaucus. So I'm confident that with persistence, things are going to get better. I wanted to talk to you about the cool thing about this grid monitoring is you can look at the distribution of any species that is in your plots. So you can develop heat maps, which I've done here for rat-tailed fescue. We can see where in the center of the restoration area is where we need to focus our efforts on that grass. And you can also look at diversity or richness of all the species in the quadrat. So native species richness, you'll also notice is low in the middle of the restoration area. So high non-native grass and low species richness of native plants. So that's our area we're going to focus on next. In conclusion, Coastal prairie restoration from perennial grass old fields is possible using an integrated suite of treatments. Direct seeding is viable and cost-effective, but you really need to take advantage of the tabula rasa once you get rid of invasive species, because it's really hard to work back into areas where you planted and then invasive species have come up. And then monitoring and adaptive management is important. And we'll just continue. Fortunately, as a national park, it's our mission to restore prairies, so we can devote staff time to this in perpetuity. So we'll just keep pulling weeds, and eventually there'll be a native grassland there. I want to thank everyone who helped us with this, and I can take questions. Okay. Yes?
Just to make sure, when you did the mantis furrows, would they broadcast onto the furrows, or did you mix the seed with the furrows?
We made furrows, and then we just scattered seed in the furrows, and then we covered them up.
So did the sparrows, they undug and ate them?
The sparrows, we came back the next day, and the furrows were completely dug up with not a single seed in them. There were a lot of sparrows here, and it just shows that, like, each restoration site has unique characteristics, and if someone says to you, this is the one way to do restoration, then they probably haven't had sparrows on their site. Yes?
The grid monitoring, did you find that it was worth the effort? And what are some alternatives to grid monitoring? What are other monitoring things you tried?
Line transect monitoring is quicker, and it's kind of the industry standard. But I like grid monitoring for large grasslands because, you know, to detect patterns, because they're hard to see on the ground. Yes?
Have you thought about using pre-emergence?
We don't have a pest track for pre-emergence, and we prefer not to use them. But I have thought about it.
Thanks, Eric. That was an expensive meal for those sparrows, for certain. I don't know if they realized that. Well, very happy to introduce our next speaker, David Banuelas. David is a first-generation PhD candidate at UC Irvine, another UC Irvine connection, with indigenous and Hispanic roots. David is passionate about restoration ecology. His research in the Kathleen Tressiter lab at UCI is focused on determining how restoration efforts affect fungal diversity. He, outside of his academic research, David is also a staff member at the Newport Bay Conservancy. David.
Awesome. Well, hello. Thank you for the introduction. My name's David, and I'm really excited to be here with you. This is a restoration site we have in Newport Beach, Southern California. Very unique restoration site. This big creek drains into the upper Newport Bay, which is Southern California's largest estuary. And the title of my presentation today is going to be about the removal of invasive woodland, and it also could help to restore ectomycorrhizal for native willows. So I'm mostly talking about the salixes today. And then before I get started, I'd like to acknowledge the Kish Nation as well as the Ashaman tribes, which traditionally washed over the lands and the rivers and estuaries that we had here in Newport Beach. And they're also the Kish Resource Management Agency. They also helped us with the actual project and permitting. They're really active in cultural resources, and there was a real great experience to have them at the actual project site and actually give us some First Peoples knowledge of the site and everything. So it was a real privilege to work with them. Okay, so I'll give you a quick overview of my project. First, I'll be talking about mycorrhizal fungi, and mycorrhizal fungi are not all the same. There's different types. There's the ones you see in the Super Mario games, and there's ones you see with just growing grasses. So I'll kind of explain that for you today. And then the next thing I'll be talking about is the really scary Brazilian pepper tree. It's not responsible for Amazon deforestation, so it's just in North America that becomes a problem. But in South America, it's a really important species for indigenous cultures, and you can make a really good wine out of it. So if you need pepper tree seeds, we have tons. The next part is going to be about the Big Canyon. And so Big Canyon is a city-owned parcel that we have in Newport Beach. And it's a really amazing spot, and it's really urban. So people walk from their homes, and you could literally walk your dog in 2 minutes. But the problem is a lot of the Brazilian pepper tree comes from the urban areas. So it's a balance there. And then I'll get into like the scientific stuff and the hypothesis and all that great stuff. And then I'll give you some results, and hopefully I leave some time for questions. And so looking forward to that. So first, here's a really good graphic. So sorry if you can't see in the back, but I'll try to explain it the best I can. So when you look at mycorrhizal fungi, the first group I'm going to be talking about is called ectomycorrhizal fungi, and then from here on out, I'll be saying ECM. So I'm not ATMs or anything like that, just ECM is ecto. So, and those are the ones that Super Mario Land kind of uses. Those have fruiting bodies, which mushrooms, but the most important part is really the below ground interactions. So you can see the blue kind of highlighted around the black roots. So really ectomycorrhizas extend the surface area of the roots and help with a multitude of things. And then the next group of mycorrhizas on the far right in the green box are buscular mycorrhizae, which only stay underground, and they actually penetrate the root cells, and so they live right inside of the plants themselves, which is contrary to the ECM. And I'm going to be calling them called AM, and I'm not talking about AM, PM now, but I'm just talking about AM, or buscular mycorrhizae. And I'll give you some more examples of AM trees and ECM trees, but AM trees, arbuscular mycorrhiza and AM is in more than 80% of plants on Earth, so it's really common. And then the main thing to remember in this slide is just that on panel D in the bottom, some trees can have dual associations with ECM and AM. And to give you like a quick overview, so blue algaberry is an AM tree. That genus around the world only recruits AM, and there's been no other studies that contradict that. And then we have the royal willow, which is the Salix genus, and the Salix genus is worldwide, has dual mycorrhiza. So if you're interested in the polar willow, let me know. There's a lot of cool stuff about it. And then a lot of tamarisk discussion today. So tamarisk actually doesn't use any mycorrhiza. So it doesn't like Super Mario, it doesn't like any of that. And it actually is an antagonist for mycorrhiza. So, and if you're thinking about soil salinity, tamarisk really makes an environment that's higher in salinity, so that's kind of bad for mycorrhiza. Maybe. We'll see later in the presentation. And then another tree that's pretty common that we have in Newport Beach is the ash tree, and that's also a dual mycorrhizal. But looking at the big bad Brazilian pepper tree, we know quite a lot about it, and it's mostly from Florida because they planted the whole state they planted with it. But on the left side are the established mechanisms that have been well known. So that's like enemy release, the physical environment, which includes disturbances, and then resource use. So old farms with nitrogen, they love that. But the thing I want to focus today on is the recruitment of beneficial soil microbes. So beneficial soil microbes, and my talk today is just mycorrhiza. So if I hear like symbiotic fungi, that includes AM and ECM, both types of mycorrhiza. And the Brazilian pepper tree, so far, we know that it does AM and ECM, so it's a dual mycorrhizal. So you have Brazilian pepper tree on two fronts attacking you, so it recruits both of them. And then, so looking at what happens with Florida, the main thing to look at is the Brazilian pepper tree really likes the glomus species of AM, so it has a particular preference for certain mycorrhiza, and it affects the diversity. So the panel with the red arrow is the Brazilian pepper tree, and then H. patens, Hemilia patens, and Biden's alba are some random plants in Florida. So, but basically from that graph is that their Brazilian pepper tree selects for specific taxa of AM, or ectomycorrhizal. And then on the right, this lengthy list, the only thing you really have to know is Russula and Chinus tuberifolius. That's like one of the common ectomycorrhizas or ECMs that it has. Where it says H patents on the bottom, they basically have no ectomycorrhiza. So this is why I came into this project thinking that the Brazilian pepper tree was going to be like this crazy super tree that had like all of these connections with symbiotic fungi. So that's basically where I got it. And then, so looking at sort of the restoration that happens with the Brazilian pepper tree, in Florida, like you can see it's almost in every county. And then in Southern California, it's mostly like Riverside, San Diego County, things like that, coastal counties. And Hawaii. Hawaii is also bad. So that's my next project. After this, I'm going to Hawaii. But looking at Florida, here's what the plant looks like in the environment. And it just it actually reproduces from like the rhizomes. So they can like, you can cut them and they'll come back. You know, it's just like the Halloween movie with Jamie Lee Curtis. It's just, another one just keeps coming, and I don't know how. But here's a mechanical removal, and you can see that they basically had to dig up all of the soil and roots to go to the limestone to actually restore the wetland. And so this project, I think it started in like 1999, and it was like 6,000 acres. So if you see the green lines and the bare lines, the green chunks those are the Brazilian pepper tree. So it was about 6,000 acres in the Everglades National Park, and it cost $20 million, and they just finished it in 2019. So yeah, so get started before, get going on that before it gets really big. Okay, and then shifting gears to my neck of the woods in Southern California, the Newport Bay estuary here in the middle, you can see where it says Newport Beach. That's mostly where we have our infestation. And the problem there with the Brazilian pepper tree is that it competes on two fronts. So it could be with coastal sage scrub, it could be in riparian communities, it could be in wetlands. I mean, it could be going in your grandma's backyard. I mean, it could be anywhere. So that's the tough thing about the Brazilian pepper tree. And then other thing that we wanted to look at was how is it affecting our native willow habitat. We have a Lisbeth vario, which is an endangered species, an endangered bird that really relies on willows and elderberries and a whole suite of native trees that we're missing because of the Brazilian pepper tree. It's taking all the real estate. And so that's the endangered species, and that it really affects endangered species because there's less nesting habitat for them, less material for them, and less habitat for them. So we really got a lot of funding based on removing the pepper tree was going to improve conditions for the endangered birds. And then also sea level rise is a component. So the Brazilian pepper tree also grows with mangroves in southern Florida, so it can grow right on the water's edge. So in Newport Beach, you can kayak and like pick Brazilian pepper trees while you're kayaking. So, but that's a problem because if sea level rise comes, the salt marsh doesn't have anywhere to go or any refuge. So getting rid of Brazilian pepper trees is like a super important issue we're dealing with in Southern California. And then, so moving on to the actual project, here's what it looks like. So that's a Brazilian pepper tree. And then you can't really even recognize the willow in this picture, but it's in there. The green arrow, it's kind of hard to see, but that's the willow that we're talking about. And then this particular project, we focused on the arroyo willow, although we have some other willows there too. And then looking at our site here, the red triangles represent places where we sampled that was all pepper tree, and then the magenta colors, those are areas of willow habitat that we sampled from. And then this is the project pre-treatment, and then after treatment, you can see like we have a lot of different habitats we included in the restoration. But the main thing to see is that the spread of the infestation in our 10-acre parcel that we have in Newport Beach. And then looking at it from above, you can see here, that's the whole invasive woodland from above. So you really can't like see anything. And like, it was also like also a problem for like urban kind of people doing things, like high school kids going in there and having fun and stuff. So it was also a hazard for like the local area. But there's a Brazilian pepper tree grove. And then there's the willows that are hanging on. And then this is about like 40% completion. So just like Florida, we had to use mechanical removal, and we used a lot of construction trucks and different contractors to help us with that. And then there's our willows there, and that palm tree is going to be gone. So And then here's the actual creek. So if you could have tell, you can even see the creek in there. You wouldn't even know there's a riparian community in there, but there's a creek there. And then there's the willow, some of the willows that we sampled from. So the area got a huge disturbance in itself. So we might have got rid of woodland, but we also impacted the native species. And we decided to leave the native willows on there because they survive shot hole borers, so we thought they were really tough. So we wanted to keep them, and a lot of arborists wanted us to remove them, and so we argued with them, and that's why I did my PhD. So I'm glad they kept them. Oh, and there's me talking about the willows. So yeah, this comes up to my next research question. I wanted to know if the Brazilian pepper tree had more symbiotic AM and ECM fungi than the native willow. Just plain and simple, just wanted to know if the invasive recruits more symbiotic fungi. And then the second thing was, I thought because you removed the Brazilian pepper tree, I was certain that the symbiotic fungi associated with the Brazilian pepper tree would increase. So those are my two predictions and research questions. And then so moving on to the results, what actually happened was in soils, here's our first graphic here. The blue squares represent native willows, and the red squares represent the Brazilian pepper tree. And the first blue square is before treatment, and then the second blue square is after treatment, and so on and so forth for the Brazilian pepper. But if you look at the first two boxes here, where it says A and B, it was actually the native willow had more richness of ectomycorrhizal, which was really surprising. But the good thing is that while there is a slight decline in the native willow habitat after you remove the Brazilian pepper tree, according to the richness of ECM, the pepper tree soils, the invasive pepper tree soils actually increased in richness of ectomycorrhizal. So it had a beneficial effect on, the restoration had a beneficial effect on it. And then, so species richness is just looking at the different types of species of ECM we have, and then species abundance looks at how much of those species are actually there. And so we also had the same thing. The salix before the treatment had a higher abundance of ECM compared to the invasive pepper tree. And then for AM species, there was an effect on the treatment. So the treatment didn't have any effect on the species richness for either plant species. But if you compared the pepper tree and the willow together, the pepper tree had more species richness of AM. So contrary to the ECM. And then there wasn't much that happened with the abundances, so nothing really occurred. There wasn't more AM or less AM after the treatment or between species. The only thing was that the Brazilian pepper tree or the invasive had a higher richness of AM fungi in soils. And then in root samples, we looked at the salix after the treatment, and it had a slight decline in richness. So that kind of goes according with the soils. So it looks like the restoration actually had a sort of a negative effect on the willows, but they're still alive, so that's good. But yeah, this just shows that there is a slight decline in the species richness of ECM. And then the abundances didn't happen. Nothing happened with the abundance at all. So there's still the same amount of mycorrhiza, but just a little less species diversity in ECM and the native willows. And then for symbiotic fungi in the roots with AM, there was a decline, but it wasn't significant. So the AMs were in really low abundances, so it's kind of hard to see in the back. They look like little lines, but basically for the AM and the root system, nothing really happened between species and after the treatment. All right, and then so for the last part of the talk, I just want to focus on ECM because I think it has the most potential for future restoration projects. So if you see on this lengthy list here, there's a whole group of different ectomycorrhizal fungi. So this winter, when it really rains, I'm going to be out there trying to find all those species and check them off. But the one I'm really excited for was called Geopora, and it's the gray kind of brownish one. That's the species, and you can see before and after, and then salix is on the left, and then the pepper tree's on the right. So you can kind of see the distribution of different species. And then in the root system too, we also have Geopora. It's the brown. So that I really thought that species had a lot going on with that. So I was really interested in it, and so we did this analysis I don't know what happened to my graph, but it got cut in half. But basically, we did a statistical analysis, and we found that the willow only had 4 ectomycorrhizal species that were only associated with the willows. And it was statistically significant, and that was Pulvinula, Angulospora, and Tomatilla, and Geopora. So Geopora is the one I was really excited about, which is OTU 16, and it was also in the root system too, as well. So basically what you get from this graph, there's 4 species that the willow only has an association with compared to the pepper tree. And so just to summarize my results really quick, the left-hand side shows all of the resilient before restoration, and the other side shows after the restoration. And the red lines indicate a negative effect, and then the green lines show a positive effect. And then black is just neutral. So basically, the invasion disrupted ECM for the mutualism of native willows. So because the Brazilian pepper trees was there was less species richness of ECM in general, and that's indicated by the top 4, like the red lines on the left side. And then AM was more associated with the pepper tree, so it had higher species richness. And you can see on the bottom panels, it has a lot more green, signaling a positive effect. But the main thing to take away is that the restoration did have a negative effect on the willows as far as species richness. So you can see on the negative lines on the top right corner, there was a negative effect. And I don't know what happened to I was supposed to say that. There it is. And so basically with my first hypothesis that the pepper tree would have more fungi, ectomycorrhizal fungi, it was only true for AM, but it wasn't true for ectomycorrhizal fungi. So I rejected that hypothesis. And then the next thing was I thought because you remove the pepper trees that it would have a positive effect and like the native symbionts, the ECM, ectomycorrhizal would increase, but it didn't increase. And I think a lot of that has to do with all the bobcats and construction trucks. It was a huge disturbance to remove all of that soil out of there. So I think in time, I think things will recover. And I think you'll have a better species richness and everything. But the good thing is that we left those willows there. So those fungal symbionts are still there, and there's willows there that are keeping the population going. So our new willow plantings hopefully will have in the future the opportunity to gain the same relationships with the ectomycorrhizal fungi. And it looks like AM is not a big factor with willows. And just to give you so in case you see this winter, this is the Geopora, what it looks like. And this is like the only iNaturalist input from San Nicolas Island. So pretty crazy. It's on the middle of an island. But this one was used with DNA sequencing. So this matched up with partially with the one that we found in Newport Beach. So Geopora is all over. So I encourage you this year to really look for it. And the really exciting thing is that Geopora, it helps a lot with soil salinity and globally. So there's a lot there. So I think this is a good ectomycorrhizal species you might want in your restoration. And I'd like to thank the Newport Bay Conservancy, Zach and Elise, Jennifer, Neil, and Emily with the lab work, and then Morgan and Soon for helping me with the field work. And I'd like to thank the Tree Seeder Lab and my sponsors.
One question.
Okay. Oh, yes.
I know you're just mostly focusing on the fungi, but for the restoration site, are you doing active restoration, and are you considering doing any inoculation of the site?
Yes. Well, we consider doing future inoculation, but where we get our plants from, they do use inoculants. So we get our stuff from Tree of Life Nursery mostly, the most locally sourced nursery we have, and then we're also doing active monitoring. So part of the funding we have like a 5-year period of monitoring, and so we still pull invasive species from there, and we're also actively maintaining what we planted there in 2020. So it's still an active restoration site, I would say very much so. Thank you.
All right, thank you. Our next speaker is Christiana Conser. Christiana has 25 years of habitat restoration experience. She's worked for HANA Resources, Plant Right, CISAC, which is the California Invasive Species Advisory Committee, River Partners, and the Golden Gate National Recreation Area. Her experience includes restoration project development and planning, grant funding, HRPs, CEQA, compliance, planting design, implementation, monitoring, data analysis, and outreach and engagement. A whole bunch of stuff. Good to have you, Christiana.
All right. Hi, everybody. End of the day on Friday. I'm going to be talking today about a project we're really excited about. We've been working in the Hidden Valley Wildlife Area in Riverside County, developing a long-term restoration project following long-term arundo removal. And I work for an environmental consulting company in Orange County called HANA Resources. We have a sister company, Nature's Image, that does restoration implementation. These are our project partners. We worked with our client, which is Riverside County Parks, who owns the property along with CDFW. And then we've done the project development and funding, and we'll do the implementation of that project. Our project partners include also the San Bernardino Valley Municipal Water District, who's also working in this area, the Santa Ana Watershed Association, who's done a lot of arundo removal over the years, and we're funded by the Wildlife Conservation Board, which has also been a really important partner in developing the project. Okay, so our goals for this project is restoring and enhancing riparian and upland habitat. So our active restoration is going to target about 600 acres. And then the entire wildlife area is about 1,500 acres. We'll be doing arundo removal on about 1,100 acres. And I'll talk a little bit about disturbance in a few minutes, but one of the goals in this is to reduce wildfire risk along this riparian corridor on the Santa Ana River, which is kind of an odd thing to have riparian corridor be fire prone, right? We also are working to increase breeding habitat for leaf-billed vireo, which was mentioned in a few talks earlier today. And there has been a correlation between reducing arundo cover and increasing breeding pairs of leaf-billed vireo on the Santa Ana River. We've been doing long-term cowbird trapping and are going to increase the scope of that trapping along the river as well to benefit leaf-billed vireo. And then increasing habitat connectivity along the Santa Ana River for large mammals. And if you're from Southern California, you know that development and encroachment is a huge problem, and so preserving wildlife corridors is really, really important for things like bobcat, mountain lion, et cetera. So we're working in the Santa Ana River watershed. It's a huge watershed. I think it's 97 stream miles. It starts up in San Bernardino Mountains, goes through Riverside County, and out through Orange County to the Pacific Ocean between Newport Beach and Huntington Beach. And I'm going to be talking really specifically about Riverside County. It's had a long-term arundo management program. So it started in about 1991 when 90% of the river was covered in arundo. Team Arundo was formed, which many people, if you've followed Calipsy over the years, you're familiar with Team Arundo. It was one of the first interagency efforts to treat arundo on kind of a regional scale. Some of the partners, and I'll list the full list in a minute, but Rivco Parks, Orange County Water District, San Bernardino Valley Municipal Water District, Santa Ana Water Association, and also the Army Corps of Engineers. And then our project partners that I mentioned before have been involved in this arundo management, since the beginning. So fast forward to now, we've had ongoing arundo management for about 31 years. We've seen some great progress. The arundo cover has been reduced to about 30%, but what we're seeing is that these areas that previously had arundo are now denuded, degraded, and they're really going to require active restoration to bring back the native vegetation. It's not happening on its own. Okay, so this is the Santa Ana River in Riverside County. There's a lot of different landowners, and I know it's hard to see the legend here, but basically it's owned by a lot of municipalities, Army Corps of Engineers, CDFW, cities, and things like that. Our site, I should have asked about the pointer. Our site is in the teal right in the middle, around Square 12 is where Hidden Valley Wildlife Area is. So how we kind of came upon this project is we sat down with all these partners and a huge map of this region and really started to look at which areas really could use active restoration to sort of get it over the hump of, the Arundo's gone, now what, increased biodiversity. And we landed on Hidden Valley because it's right in the middle there, and it's a very important piece of habitat that connects this watershed. This is a more comprehensive list of the partners and the people that have been working in this region of the Santa Ana River. So CDFW, Coastal Conservancy, 2 resource conservation districts, Inland Empire and Riverside Corona, the flood control districts, water districts, and federal agencies like Army Corps, who's working on Prado Dam and the Alcoa Dike project just downstream from us. U.S. Fish and Wildlife and USGS, and then all of this is pretty much within the Western Riverside County Regional Conservation Authority, so it's part of that Riverside County MSHCP. Okay, our project is located where the star is, and I like this showing how how the river's coming through. But basically, it's winding around through Riverside County, and then on the bottom left, where you see the green, that's where Prado Dam is. So we're sort of right in the middle of the San Bernardino Mountains and the dam. This is Hidden Valley Wildlife Area. It's about 1,500 acres total. Our project is about 1,200 acres in total, and the reason it's less than the total site is that there are some mitigation areas for some of the partners that are outside the scope of our project. So there's still habitat on the southern side of the project. It's being managed by San Bernardino Valley Municipal Water District. Okay. So we looked at, Cal IPSI did a statewide project where they looked at Arundo distribution and impact, and this is what Hidden Valley looked like in about 2011. More than 50% Arundo, and this is after 20 years of Arundo management at that point. So obviously there's a lot of work to do. There's ongoing work that's still going on there. Riverside County Parks has been tasked with eradicating Arundo on all of its properties and restoring those degraded habitats for native plants and other species. So this is an example of one of the areas where we've been removing Arundo. So on the left-hand side, you can see giant reed is pretty much all the bright green in there. SAWA, Santa Ana Watershed Association, got a Prop 50 grant and did 6 years of work out there. And on the right-hand side, you can see the results of that, which is the Arundo's gone, the willows are hanging in there, but there's still a lot of bare ground in between. So HANA went out in 2020 with a drone, and we mapped the site and created this orthomosaic image. We also have a plant recognition software. It's an AI that we've developed and have a patent for, and it can map and recognize Arundo from aerial photographs. So the Arundo that's currently on the site is in red, so you can see it on the west end and the east end, and then little patches all the way around. About 41 acres are left. Okay, so this project is actually really interesting because the Santa Ana River has had a dramatic shift in its disturbance regime in the last few years, and a lot of that has to do with Arundo. So this is a wild river. Doesn't have levees on it like they do up in Northern California, right? Which is what I'm used to with river partners. And so prior to the building of Prado Dam, which is on the border of Orange and Riverside counties, we had really high flows on this river. So pre-dam, 320,000 CFS was a high flow. After the dam was built, the high flow went down to 100,000 CFS. And then Seven Oaks Dam was built in the year 2000 up in the mountains in San Bernardino. And since that dam has been built, the high flow has gone down to 49,100 CFS, which roughly translates to a 50-year flood event. This is what Hidden Valley looks like at a 50-year flood event. Most of the site is flooded, and the colors are kind of backwards on this. So the yellow is deep water, so you can see the river channel is in that deep water, 10 to 12 feet. But the rest of the site is pretty much covered in between 2 and 8 feet of water at this flow event. So challenges, right? So this is one of the reasons that so much arundo has been washing down this rivershed and just continually replenishing the arundo that's there. So it just keeps coming in. You remove it, it comes back. So now we have this type conversion where we have arundo moved in, and instead of the dominant disturbance being flooding, it's now fire. So when we first started working on this project, this was one of the first wildfires that we saw burn there, but many, many burn there every year. So this is on the west end of the site. This was in March 2020 on the left-hand side, in an area that SAWA had been managing for arundo for years. This is kind of roughly in the same region as that Prop 50 project. So it burned the arundo, and then we came back in July, and all of the background there, that bright green, is all the arundo coming back from rhizomes that we thought were dead. So when we went out and looked at it on the ground, you know, it sort of looks a little promising. Is it burned? Is it dead? Will it come back? And then you start seeing where that red circle is. It will come back, right? So it's not actually dead. So we got out there, we continued to kind of remove it. So the biggest problem is arundo. There are other problems there as well. We did some vegetation mapping out there, and all the earth tones are different dominated non-native communities. So everything from eucalyptus, tree of heaven, perennial pepperweed, poison hemlock, castor bean, things like that. A lot of non-native grasslands. Fortunately, we have in the green, we still have riparian habitat there. So our idea is to remove the non-natives and build on the native communities there. Okay, so here's some of the tasks that we're taking on for this project. We're going to remove the remaining arundo stands and continue to treat those resprouts and new infestations as they crop up. We're going to remove other high priority invasive plants. We can't tackle them all. So we're focusing on castor bean, salt cedar, tree tobacco, poison hemlock, and perennial pepperweed. We're going to restore and enhance riparian and upland habitat. So active restoration will mostly be focused in areas where large, dense stands of arundo were removed, and that's what this photograph is kind of showing. So we've removed the arundo, and now we have this bare, kind of degraded land. And then passive restoration or enhancement will basically be in areas where there's small, less dense stands of arundo that were removed, and we can basically count on the native vegetation to fill in over time. Okay, some of the benefits of the project are going to be increasing connectivity. As I said, this is a really important wildlife corridor. Increase riparian and upland habitat for wildlife, including leaf-spells vireo. And then also working on putting in some fuel breaks and doing fuel reduction in some of these areas so we can hopefully prevent these wildfires from spreading across the site and also into the adjacent urban area. The project's going to be laid out in 5 phases. So we're completing phase 1 right now, which is environmental compliance, and then we will have 4 phases of active restoration, which I'll go into next. So phase 1 was our compliance. We're completing our CEQA compliance via SERP, which is the new statutory exemption for restoration projects, the Cutting the Green Tape Initiative, which came along right in the nick of time for this project. We're also doing a lot of stakeholder and public outreach, so we'll be posting our habitat restoration plan online in the next month, so if you want to comment, please check it out. We've done a lot of technical studies out there, which I don't have time to go into, but just sort of describing the site and baseline conditions. We've created a habitat restoration plan, and we're completing permitting, which is great when you have a lot of partners who have programmatic agreements, and you can sort of hitch your wagon to work that's already being done. Okay, now I'm going to show you just quickly maps of our different phases of restoration. So phase 2 will be the next phase. We're going to focus on finishing the arundo biomass removal and clearing the rest of it out, and then doing what we're calling a mule fat scrub restoration, which is really one of our partners from the San Bernardino Valley Municipal Water District did his dissertation there and found that really high density mule fat plantings can shade out arundo resprouts. So we're going to try this technique here. In the areas with red, we'll do really high density mule fat, and the other areas we'll do a mix of mule fat and arroyo, Goodings, black willow, and red willow. And these phases will be 5 years each. Phase 3 will come in next, and we're going to focus on riparian communities, so southern cottonwood willow forest and willow riparian. And I have the species or the planting palettes here. They're basically in order of dominance, and so they'll be diverse, but mostly focusing on willows and cottonwoods and things like that. Most of these plantings will also be occurring in areas that don't have high flood flows. Phase 4 is going to be riparian scrub and River Sidian alluvial fan sage scrub, which is a community that is very rare in Southern California, so we're going to be working to restore that. And these communities are even more diverse than riparian, so it's really important to have them here for food resources and things like that for wildlife. And then our final phase will be a coastal sage scrub cactus restoration, and you can see it's sort of on the margins of the site. And we'll be using things like valley cholla and coast prickly pear to create fire breaks along the site and also from escaping the site into adjacent urban areas, and then hopefully providing cactus wren habitat as well, which is not present on the site, but there's a movement for promoting that type of habitat in this area. So this is what the whole thing looks like all together. It's a total of 608 acres of restoration, and each phase we kind of tried to parse out to be somewhat equal in size. And then just in conclusion, so these long-term control efforts like Team Arundo have been really successful, but we need to do more. And so efforts should also include high-priority invasive plants. And then we're finding in large areas where Arundo's been removed, we need to do active restoration. Okay. So this is myself and my partner, Dustin McLean at Rivco Parks. We basically have been developing different types of projects around the state and developing them with partners and getting funding. So thank you.
Time for a question.
Oh, sorry, yes. Who had their hand raised? Sorry, I didn't see it.
I have a question.
Yes.
I'm doing actually a lot of arundo removal up here in Salado, Willow counties, and again, working kind of on a watershed scale. And I was curious about your removal, and so it sounds like you're actually removing the biomass as well. So again, we're working right on that in the riparian all the time. I imagine you're going in initially with, from my partner side, we were actually removing the biomass and shipping it. What are you doing?
Yeah, so we go in with a barca, which is a large kind of masticator. It grinds down the arundo, but then they do actually do cut stump treatments, so they'll spray afterwards and try to get really good control. This will happen for a couple years. The whole site's going to have constant just arundo sort of patrol to go around and see what's resprouting, because it really does take a lot of treatment to get those rhizomes to exhaust all their energy and die. But we'll be chipping it on site, really small, so it doesn't resprout from that. But yeah, they have a lot of experience and have been pretty successful with their formulary. Yeah.
What kind of energy is there available to treat the arundo upstream so you don't have to keep on fighting the same thing?
Right. Well, yeah, that's sort of, there's a very regional effort on the Santa Ana River, so we're all kind of working in coordination with each other. And making progress, but slow progress, yeah.
Unfortunately, we need to get
all right.
off, and we'll give you guys for talking.
Sure, thank you.
All right, thanks, Christiana. Okay, this is the one speaker I don't know by face. Are you?
I'm Caitlin.
Are you Caitlin? Good,
Yeah.
great. Okay, that's good to know. Our next speaker is Caitlin Kreutz, and Caitlin is an environmental resource coordinator at the Rancho Santa Fe Association. She manages over 200 acres of biologically diverse, privately held open space in the north section of San Diego County, and she has raised quite a bit of money over the last few years for invasive species removal and habitat restoration. So please.
First of all, I'm super impressed you said my last name right. Thank you. Hi, I'm Caitlin, and I'm taking a bit of a different track today. We've seen a lot of technical, really very interesting projects, but I wanted to tell you kind of a community success story. And I work for the oldest HOA in California. It's called the Rancho Santa Fe Association, and I have a background in biology and horticulture, and so I came to this job, and I was working in the Parks and Recreation Department doing other things, although I have a background in this. And I got this little note on my desk from the CNPS San Diego chapter that they wanted to talk to me about some weeds. And this is kind of the origin of our story here. And we, like the last presenter, have a riparian corridor that has been plagued by fire. And so in 2007, we had the Witch Creek fire that came through where it says Arroyo Preserve in the red, and then another fire in 2014 that came through with the orange. And we had a lot of property loss and damage. So a group of homeowners got together. They met with a colleague, Jonathan Snapcook, who is here from Partners for Fish and Wildlife, and they wanted to get together and do something about it. So this is a grassroots effort, and it's taken over 40 private partners. We have 40 right of entry agreements with private landowners. We only got chased off one property, so that's good. And we started looking and getting funding, and we were able to get, we've raised about $700,000, which I'm hoping our homeowners association is going to be matching soon. And we've been able to do a lot of work in here. So we've received funding from the State Coastal Commission, Prop 1, Prop 68 with the Urban Corps, Fire Safe Council. We even got a couple grants from REI in Patagonia. So it's been a collaborative effort raising that money. And our dream team here, we have Bob Burns. He's right here in the front as well. He is the Habitat Restoration Committee co-chair for our CNPS San Diego chapter. And our other colleague, Arne Johansson, who's not here today, is also part of that chapter. He's also a co-chair. And these guys have contributed to over 5,000 hours of volunteer efforts, which is incredible. They've done an amazing job. We could not have done it without them. Yes, please applause. It's been very inspiring working with Bob and seeing his passion. We also work with the San Dieguito River Valley Conservancy, and we get our permits through them so that we can do work here in the River Valley. Our permits don't allow us to use anything with tracks or wheels, and so we have a little bit of a different approach to how we do restoration, which I'll go into in a minute. And then Jonathan Snapcook, he was like I said, he's with Partners for Fish and Wildlife, and he was able to get the first funding for this project that really started the initiative. So it's great to have him here. Our target species for removal, they're the usual suspects. We have red gum eucalyptus. In our case, in Rancho Santa Fe, in the early 1900s, the Santa Fe Railroad began planting these trees for railroad ties. It was an abysmal failure, but In 1914, 3,000 acres of red gum eucalyptus was planted in this area, and it's since then just invaded all of our riparian communities, and it's a big problem. The picture of the red gum is actually our river valley before we got started, and you can see it's quite prevalent. We also have Canary Island date palm, Mexican fan palm, arundo, tamarisk, and pampas grass. We also have other things like castor bean, acacia, and some other nasty ones that we include in that mix. So here's our vegetation map. You can see the predominant tan color, that's eucalyptus. And it's just absolutely choking out this riparian corridor. And this is what, fueled the fire that came through here, the two fires. These trees are much larger than the native trees. They cast embers up to a mile or two away. They have volatile oils, and they're incredibly dangerous. So our efforts are fire risk mitigation and habitat restoration. We use something different. We call it the Bradley method. It's been around since the 70s. But this is a method of weed control that was developed in Australia. And the basis of this method is native species' ability to recolonize by tipping the ecological balance away from the invasive weeds towards native plants. And the method consists of invasive removal without replanting is in the methodology, although we do some prescribed seeding and cuttings. And after that, the vegetation, after the weeding, each area will be promptly re-inhabited and stabilized by the regeneration of native plants. And we've seen amazing native plant recruitment in this area. It's quite phenomenal. And we really don't damage too much of the understory when we're doing a lot of this. We try to take out things as carefully as possible. So we're keeping, the native understory intact as much as we can. And so, instead of just kind of clear cutting an area and disturbing the soil in the native plant populations, we try to keep that intact. We work from the strongholds and then kind of work our way out. So basically the three takeaways is prevent deterioration of good areas, improve the next best, hold the advantage gained, and then cautiously move into really bad areas. So we find this to be effective for us, and I have some photos that will show all this native recruitment, and it's quite impressive, I think. So here's a picture of before this one area. You can see this is typical of our river valley, just a bunch of junk in here. And so here's a photo of afterwards. This is right afterwards. And you can see it's like, oh my gosh, there's a river in there and it looks lovely. And another one, here's our before pictures. We've got just tons of eucalyptus. Oh, and there has been efforts made upstream in the Del Dios Gorge area, and that has been restored. So we are moving downstream. And here's after. Here's a picture of it. It's beautiful. It's lush. It is filled with a couple of our native willows, mule fat, baccharis. We've got Platanus racemosa and cottonwood trees, and we do a lot of mule fat cuttings as well as some seed distribution. But we are aiming to restore the biological, ecological function of this river valley. Here's another after picture. You can see there's not one eucalyptus in sight. It's music to my ears. Here's a picture of some of our crew working on arundo removal. We have similar methods to the speaker that just came before me on how we manage that. I'd love to compare notes, so afterwards we'll have to chat. Here is dear Bob in a sea of pampas grass. He has annihilated every last one of those. So they're dead. Here is me slaying some tamarisk. And we do use herbicides. We use different types depending on what we're using it for. We use glyphosate mainly and also triclopyr. And for the eucalyptus, we usually do, and anything that's woody, we do a cut stump treatment. So we'll cut it down, drill into these are pretty small, but we'll drill into the cambium layer and use a dropper to put the herbicide in these holes. And that actually helps kill them and keep them dead. We do have to do follow-up treatments from time to time, but this kills the vast majority of them. And then we do, with the arundo, we do cut it down, wait for it to grow a little bit, and then spray it. And we have to do that several times. It's quite noxious. And the tamarisk also needs more than one treatment. Like you said earlier, it really wants to be here. So we try to get, we've taken out thousands and thousands of these eucalyptus, and every single last one of them is drilled by our team. So we're lucky to have such great volunteers. We do a lot of private landowner outreach, and this, there's a big learning curve when you're talking to people about this. We kind of lead with the fire thing, because there's a lot of property damage, and we talk about the importance of native vegetation and habitat. And they eventually come around, but in Rancho Santa Fe, if you're familiar with the area, it's a little difficult. I've become a great negotiator, let's just say that. So, I think there's a lot of people who have come around, seen our work, decided to sign on, and we've been really successful in that. So it's been quite a good experience. Since we're competing with rare plants over there, I thought I would discuss a couple of the rare plants that are found in our preserve. And we've got a number of them. I'm just going to kind of breeze over this, but the rating scale. We have a couple 1B1s, which are quite rare, threatened, and endangered, and then we have a couple 2s, 3s, and 4s mixed in. So the first one is a 1B1, Nettles scrub oak, Quercus dumosa. This is a great little shrubby tree, and I live in Encinitas, which is in North County, San Diego, and Encinitas means little oak, and it was named after this species. So it's great to see it. We have a good stronghold, and it's very slow growing, so we're lucky to be able to preserve our stands of this. Our second 1B1 is Encinitas baccharis. We have this in our preserve. There's only about 15 populations of this left, so we're doing our best to conserve that. The poor thing is just terribly ugly, so people, don't care too much about it, but I care. So I'm doing my best to make sure that this continues to spread in the preserve. We have also summer holly, the Comaristophilis. This is a 1B2. We have a couple of these. This is kind of a rare subspecies of this, and we have a couple populations of it as well. And then we have California Adelphia californica. This one is rare in San Diego, but it is more abundant down in Baja region. But this is an unusual. It has photosynthetic green stems, and it's very prevalent in the preserve. So we've got a lot of it, which is great. We also have this volcanic outcropping at the preserve, and it's covered with Phaerocactus viridescens, which is really cool. It's a little further east than you would maybe find some of the other populations. Unfortunately, we do get some poaching situations down there, and so luckily the preserve is private. It's not open to the public, so we have a little bit better way to monitor that. And I'll have to say, since we are private property in an affluent community, it is a little difficult for us to find funding. So we don't have public lands. We can't get those big grants, but we do find some agencies that are able to do that, like the Natural Resources Conservation Service, and they've been absolutely amazing in helping fund this. One of our big understory shrubs that we have is the San Diego marsh elder, Iva haciana. And this is a 2B2. It's very locally abundant in the area, and this regenerates quite quickly when we give it the space it needs. So that's pretty nice, and it smells very, very pretty too. We have wart stem ceanothus, Ceanothus verrucosus. This is a 2B2. It's in the area, and apparently you can put the flowers in the water, and they kind of make some suds. So that was an ethnobotanical use of this, which is pretty cool. We also have another ugly plant, but I appreciate it. Selaginella, ashy spike moss. This is very sensitive to soil disturbance, and it needs clayey soil. So we have a couple lenses in the preserve where this is abundant, and it's similar to the resurrection Selaginella, where it comes back in the spring very rapidly when it receives some rain. So that's fun to see. We have a spiny rush, Juncus acutus leopoldii. This is a 4.2. This also comes back very rapidly in our area after we've taken out the invasive species. It's very spiky. We think we've been poked by it hundreds and hundreds of times when we're out there. It hurts. But it's a very important plant that grows in our area. We also have San Diego sagewort, Artemisia palmeri. So apparently, according to Calscape, David Bowie ate this during his recording of his album Low, which is just weird, but interesting. So then I kind of was doing some more research, and I realized the Nature Collective kind of clapped back at that and was like, hey, he was in France during that. So I think he was actually using the Artemisia absinthius, which is the absinthe one. So that would be my idea. And we have a couple endangered and threatened bird species. We also have the least Bell's vireo in our area. We also have California gnatcatcher, which is my spirit animal because it is the cutest, and when it makes its little cheep, it sounds super annoyed. It's just like me, me. It's the best. So I love hearing it out there. It just makes me so happy. Also, our project area upstream has recently, I just got in my email this week that they're having occurrences of ridgeway rail. So we're really excited to hear that because I'm sure it's in our preserve as well, and we'll be listening very closely when we're out with our Audubon doing our monitoring. So that's pretty exciting. Like I said, we do lots of wildlife monitoring. We do bird surveys. We also do camera trapping with the San Diego tracking team. These are some of my favorite images that we have got with our camera, but we also have long-tailed weasel. I found a badger den, which is super cool, and other interesting critters. And we are, I kind of left out a lot of the statistical analysis of all of this, but we have seen overall an increase in mammal populations. And I also do remote sensing and monitoring and vegetation analysis, but I wanted to try to keep this a little light because it's a Friday afternoon, everybody's hungry, I'm sure. And we also do herpetology surveys, which is really fun. We find all sorts of cool critters. We've had our cover boards out for about 6 years, so they're very productive at this point. And the bottom there is a Gilbert skink, which is pretty neat. I had never seen one before. And then just to conclude, our project was in the Flora magazine in winter 2019. You can find that online. So if you're interested in learning a little bit more about our approach and the things that we've done to get to where we are, please check that out. And then here is our contact information if you're interested. I would love to talk to people who work for HOAs. It's like the HOA is kind of, everybody in California has an HOA. It's kind of an abusive relationship, and it can be really difficult to get things done. So I would love to talk to people who have challenges, especially if they have private property that they need to do a restoration on. I'm happy to help you with some techniques. And I think my time's up, so I don't know if I have time for a question. Let me know. Okay.
Yes, one question.
I've got one question if anybody's got one. Yes.
I understood you say you couldn't use willow on grass vehicles. How did you dispose of the
That's a good question. We would fill dumpsters and dumpsters and dumpsters and dumpsters full of this stuff, and it went to go to biochar. We also do a lot of chipping on site. In the community that I work, we have 60 miles of equestrian trails, so we do a lot of chipping and reuse it that way. So mostly biochar and chipping. That's a good question. Also, the topography in one of our areas is going to require some helicopter extraction, so we have to get a little creative. We also partner with a local woodworking association, and they'll come make things, and we try to be as creative as possible when getting rid of it. But good question, thank you. Okay.
Thank you, Caitlin, and especially thank you for ending us on this note of reminding us all of why we're doing this, showing us these beautiful pictures of the wildlife and the plants and the habitats that we're trying to protect. So appreciate you guys being here. Any last words, Doug?
Well done.
All right, well, thanks, and we'll see you tonight.